Okay. Good afternoon to all the companies. This is the third days of the winter school of the University of Padua, and I just share the screen to remember the scheduling of this day. The general topic is data science for omics sciences. Apologies for cross-wording. The general topic of the session will be addressed by Luca Vedovelli, a postdoc fellow in biostatistics at the University of Padua. And we have a free intervention of about 40 minutes by Giovanna Chiorino and Paola Ostano from the Cancer Genomics Laboratory of the Fondazione Edo and Elva Tempia in Piedmont, in Biella, on Omics Technologies for Cancer Detection and Prediction of Prognosis. Then we have an intervention by Barbara Pioselli and Maria Laura Faietti from the Chiesi Pharmaceutics, researchers in science, in preclinical research and development and analytics. This intervention this talk is mainly on preclinical proteomics, theory and application towards translational medicine. The last intervention is from Manuela Simonato, a postdoctoral fellow in Metabolomics and Mass Spectrometry here in the University of Padua, the Foundation Istituto di Ricerca Pediatrica Città della Speranza, and is again on metabolomics application in medical and biological sciences. We have a floor discussion after the free intervention, but if there are some burning question during the presentation, we can have a few minutes between each of the presentation. Okay, this all what I have to say, and I invite Luca to give our topic introduction.
I try to share the screen. Okay, can you see?
Yes.
Okay.
Yes, it's fine.
So for this introduction, I will introduce the argument of omics data and omics studies, and in the biological and medical fields. And for this introduction, since our next speaker will go deep into the single omics technique, I will give you a glimpse on how integrating omics data can unveil the biological processes. So it means that we will consider all the omics together in a single study, and to see the pros and cons that can be drawn from this kind of data. So omics approaches are techniques that try to obtain as much information as possible from a single sample, exploring a whole class of molecules without selecting a specific target. From DNA from genomics to metabolites for metabolomics, omics approaches try to have a picture as large as possible of the roles and interplays coming from molecules with a related function. The arrow that you see in the picture on the left represents the transition from genotype, that is the last time variable of the omics set of techniques, to the actual phenotype that constantly changes in a matter of minutes. As in the clip here in the right, the orange thread represents a disease or a physiological condition that spans through different levels with their own conformation and complexity. Omics sciences try to describe these levels, while multi-omics integration try to unveil the interplay among the levels. Omics applications were initially sold as an extraordinary tool to enable personalized medicine, personalized to a point that each individual could have been characterized to the smallest modification of a molecule in our body. While this is actually quite cool to think and sell, over the years, the question if this approach could be useful started to rise from a whisper to a huge scream. Is it really feasible to tailor a specific therapy to a single disease of a single person? In my opinion, the most powerful tool that the combination of omics sciences and data that they could give is the level of detail, defining the very basic trait of molecular processes while keeping the large picture intact. Is what the omics sciences are theoretically perfectly capable of offer us. The ultimate goal is not only to personalize the treatment of a disease in a single person that is a consequence of the process, but to understand the causal origin of a disease. In other words, to use many examples to derive a global rule. As an example of the complexity omics and multi-omics have to face for causal interpretation, let's take lactate as an example. Lactate is a simple cellular endogenous metabolite. It's biosynthesized enzymatically from pyruvate, another metabolite, with the help of lactate dehydrogenase. Here is LDHA, that is a catalytic protein. In turn, lactate dehydrogenase can interact with several known and unknown proteins through protein-protein interaction to regulate their function, and itself it's subjected to diverse post-translational modification that here are represented with PTMs. These PTMs regulates its catalytic function. Lactate measurement through techniques such as in vivo brain imaging in human and other model animals can generate a lactate spatial distribution. Moreover, gut microbiome via lactobacillus and other microbes can synthesize lactate and release it into human physiological system to contribute to lactate levels. Lactate biosynthesis regulation can be due to various levels of changes that are depicted here. Genetics via SNPs, transcriptomic, post-transcriptomic, like miRNA, and epigenetics, like DNA methylation on LDHA gene. Though this is one of the well-studied set of multi-omics interaction, but one can expect more complex and unknown interaction while integrating multi-omics datasets. The state of the art of the main omics techniques that are genomics, transcriptomics, and proteomics and metabolomics. I made a summary here dividing for status, mature or maturing, application that are already available, and application in development. Genome and exome sequences are the omics study that are more mature. For example, in the case of Mendelian diseases, scientists are turning to exome and genome sequencing to find the causative mutation of their disease, which has proven successfully in 25–50% of cases previously not solved by targeted gene panels. In addition, RNA sequencing could add an estimate to 10 to 35% more diagnosis, including information on cryptic splicing sites that could have been not detected in exome sequencing because they were present in introns or neglected in the whole genome sequencing because not predicted to cause splicing. I put genome-wide association studies in the development field because it's true that it has been successful in identifying risk loci for diseases. However, in many cases, the causal variant or gene is not identified. Proteomics used to quantify proteins in multiple samples types using both untargeted and targeted approaches. Recent developments in mass spectrometry have dramatically increased the sensitivity while decreasing the amount of sample required for high throughput analysis, and now allow for detection of minimal differences in proteins abundances, identification of post-translational modification, and other applications from a wide range of samples and tissues. Metabolites are often the end products of a complex biochemical cascades that can link the genome, transcriptome, and proteome to the phenotype, providing an important key tool for discovery of the genetic basis of metabolic variation. Metabolomics can be used to determine the relative and absolute amount of sugars, lipids, amino acids, organic acid, nucleotides, steroids, drugs, and also in environmental constituents from a wide range also here of samples types, including primary cells, cell lines, tissues, biofluids, and also entire organisms, and diverse geoclimatic environments also. These technologies have contributed to medical advances by their own, but each technology individually cannot capture the entire biological complexity of most human diseases. Multi-omics aims to identify molecular markers associated with biological processes by revealing the regulatory units across diverse omics layers. Here on the right, you see a novel 2022 simulation of a minimal cell done by the NVIDIA team. And you can see also from this that it's really a minimal example of a cell, how complex is the life inside the cell. And this is what omics sciences try to unveil. So the typical workflow starts from the genotype and goes through all these processes through the phenotype. That is the ultimate goal. So the main omics are genomics, transcriptomics, proteomics, and metabolomics, as I said before, that can be added with some clinical and imaging data sets. Other smaller omics like macrobiomics, fluxomics, etc. All the data are processed through machine learning, deep learning, or classical statistical approaches. The omics are integrated, and this one will cause the description of some novel networks, novel pathways, and also model predictions, prediction of health and disease, and biomarker discovery. So a simple integration, a minimal example of these technologies that independently identify different aspects of the same biological process and thus can validate each other. For instance, RNA sequences can internally replicate exonics variants identified through whole genome or whole exome sequences, whereas proteomics expression can validate the expression from RNA sequencing. Finally, metabolomics can detect and quantify the activity of proteins. The type of studies multi-omic approaches can broadly can be categorized are supervised classification tasks that includes discrete outcomes such as disease control status, prediction, tasks like a continuous outcome like survival or pain score. Or a second type could be exploratory, unsupervised clustering, disease subtype discovery, and relationship-based analysis, for example, correlation covariance and network models. Ideally, different technologies would be combined both to help diagnose disease and to create a holistic picture of human phenotypes and disease. However, implementation of multi-omics data introduces new informatics and interpretation challenges. Specifically, novel analytical and statistical methods are needed for combining disparate data sets, as well as standardized quality control metrics. Additionally, the field must address challenges in the interpretation of molecular events and accordingly their actionability and whether they can guide therapeutics and clinical care. Regarding data analysis, the first step is always to make sure you gather data have enough quality to trust them. And data wrangling is the first fundamental steps to do. What is data wrangling? Data wrangling is also referred as data munging. It includes some various levels of transformation and mapping of data that is critical to the multi-omics field. Transformation is accomplished by data scaling, normalization, imputation that help harmonize the different omics data together. One option from drawing inference from this data involves pairwise analysis of data sets, mounting evidence to support the signal. However, analyzing 3 or more data sets simultaneously requires more sophisticated multidimensional methods, such as Bayesian methods, neural networks, or dimensionality reduction. This is further complicated by the fact that the various omics data types are fundamentally different. For instance, genetic variation are discrete and static, whereas RNA sequencing measurement are continuous and can provide longitudinal information as long as proteomics and metabolomics. So the increasing reliance of computational methods necessitates systematic evaluation that is called benchmarking of the omics data analysis tools and methods. The key challenges in omics scale benchmarking of computational tools include the acquisition of a gold standard datasets that provides unbiased, the unbiased ground truth, incorporating new methods for establishing benchmarks as they are published, and thus providing continuous and extendable benchmarks and ensuring reproducibility in the context of the increasing complexity of the software involved. For example, the Cancer Genome Atlas, TCGA, which includes genomic, epigenomic, transcriptomic, proteomic, and clinical data from 32 cancers to date, is a landmark dataset for multi-omics method development, and a recent study reveals that references to TCGA are enriched in the multi-omics computational method articles compared to other article types. So it is becoming de facto a gold standard for multi-omics research. The largest hurdle for any omics dataset remains making sense of the data. One major objective of multi-dimensional omics approaches is biomarker discovery, no matter from which omics layer they are key molecules, they are derived. Sensitivity and specificity of molecular biomarkers are essential for usefulness in biomedical research and clinical translation of the findings. Interpretation and curation of complex multi-layered networks is challenging, computationally and in time. Requires detailed knowledge of biological system being studied. Studies using an integrated omics approach without applying biological knowledge of the system frequently end with the nomination of key molecules and networks for hypothesis testing that are not biologically relevant, because validation of the key molecules, inclusion of validation cohorts and networks, for example, gene, proteins, or metabolites, is time-consuming and often challenging. So biologically informed nomination of candidates is essential. In the light of this, a more technical issue in omics data analysis is the incontrovertible desire of making sense of the data, demanding to the computer the burden of extracting the most useful variables in the dataset. This is called feature selection. If it's not conducted properly, it's a very bad idea. So bad that you can risk to give away all the information and money you spent because the model you apply is it's unreliable. As proven multiple times, but one of the best statisticians alive that we had the honor to have here at this Windows School on Monday, Professor Frank Harrell, responding to the very good question of our PhD student Silvano Salaris, remarks that feature selection, when you have more variables than cases, is known to be unreliable, but most biomedical researchers are not aware of the magnitude of the problem. They assume, for example, that settings a false discovery rate makes the results reliable, forgetting about the false negative rate, and decades of research showing unreliability of stepwise variable selection, even when the number of variables to select are low. Professor Harrell suggests to maintain the complexity of the data using different visualization techniques and then apply the field knowledge to identify what could be good candidates to test further. The process is, as you can imagine, astronomically longer and tedious of clicking a button and letting the computer to do all the job. But the probability of doing something wrong with the quicker option is so high that mistakes are almost sure to happen. Regarding clinical application of omics studies, we have to consider that most integrative models have been reported and published in research settings. For clinical adoption of any technology to occur, high specificity and sensitivity are required both in detection and interpretation. Clinical guidelines must be established to ensure accuracy and efficacy, and test to show non-inferiority and cost-effectiveness against the standard practice or the already adopted test must be performed. Nonetheless, omic profiling can be a very effective way of detecting large-scale or pathway level alteration, cheaper and they are cheaper and more often comprehensive than performing thousands of individual tests. And longitudinal profiling can show patients' specific trends and add statistical support through repeated measurement. So the difference is to try to catch a fish throwing a net or to search for a single fish with different baits. Omics techniques rely on references, gene sequences, protein sequences, and assembly, also metabolized structure. For detection and annotation, that is to say, associate a name to a gene, a protein, or a metabolite, or a biological process, every single entry on the database has to be manually derived, and carefully checked. Artificial intelligence could help in this tedious task. For example, one of the latest example is AlphaFold by a subsidiary of Google. AlphaFold database, it's free and provides open access to protein structure prediction for the human proteome, and its efficacy and accuracy is the highest possible to date. So if you are interested and want to play a bit with multi-omics integration, here are some freely available tools to analyze your data, or simply try some example available directly on the tool website. Mixomics is the most famous one, but also the other are very interesting. And if you want to know more, here there are 3 reviews that can give you a glimpse of what's going on omics sciences, and particularly on omics integration.
Okay, many thanks, Luca. And also thanks to be on time in your presentation. I don't have any urgent question, so I switch to our first presentation. So I invite Giovanna and Paola for their talk on omics technologies for cancer detection and prediction of prognosis.
Thank you very much, Annibale, for this invitation, and good afternoon to everybody. So me and Paola, we come from Fondazione Edo at Elva Tempia in Biella. That is a non-profit organization involved in cancer prevention, cancer care, and also cancer research and diagnostics. So the Cancer Genomics Laboratory has 2 complementary souls. One is devoted to experimental laboratory work, and the other one to bioinformatic data analysis, but they are strictly interconnected. And the starting point of our research projects is the analysis of cancer cells from cancer patients that may have a similar tumor but different outcomes, and so we want to compare the 2 groups. Or, for example, cancer cells compared to normal cells. We use different omics technologies, and the final aim is to identify novel drug targets for cancer, improve the current therapies, but also discover novel diagnostic, prognostic, or predictive biomarkers, with the final goal to improve the therapeutic outcome of patients. But we analyze not only cancer cells from tissue, we also analyze a liquid biopsy. That is a precious source of genetic material, genetic and metabolomic material that can be analyzed in a non-invasive way to determine, to identify novel biomarkers. And during my talk, I will speak in particular about circulating microRNAs. So this is these are the most frequently diagnosed tumors in Italy that were diagnosed in 2020, and our research is focused on mainly on these principal tumors. For example, in breast cancer, using different omics technologies, colorectal cancer, in particular in collaboration with the Regina Elena Institute in Rome, and lung cancer with the San Martino Hospital in Genova. And we are focused mainly on exosomal microRNAs to predict response to immunotherapy. But the main tumor we are focusing is prostate cancer, and we will speak about it. I will speak about the microRNA profiling of the circulating microRNA, and Paola will focus more on gene expression profiling from cancer tissue. Then I will speak also about oropharynx tumor, and Paola about skin squamous cell carcinoma. Another cancer that we study in our lab is also melanoma. So let's start from oropharyngeal cancer. I will show you the results of recent work. Oropharyngeal tumor is a subtype of head and neck squamous cell carcinoma, and human papillomavirus infection is the most important risk factor for this kind of tumor. And the majority of HPV positive oropharyngeal cancer have a good prognosis, but some of them are more aggressive, and the survival is more similar to HPV negative tumors that usually occur in older people, are more related to smoke and alcohol, and have a worse prognosis. So in this kind of these tumors do not respond always to the standard therapy, that is chemotherapy with cisplatin. So there is a need to find novel prognostic biomarker for HPV negative tumors, and also novel therapeutic targets. So we analyzed 4 independent cohorts with more than 5 years of follow-up by transcriptomic profiling. So one was an internal cohort. Another one was retrieved from the Cancer Genome Atlas by selecting only oropharyngeal tumors within the dataset of head and neck squamous cell carcinoma uploaded on TCGA. And 2 other datasets were retrieved from the Gene Expression Omnibus, that is a public repository for especially transcriptomics, but also other omics data. So we compared the HPV positive with the HPV negative tumors. We were able to identify some molecular processes that were overrepresented within one or the other class. But the main goal was to combine these 4 datasets in order to find a robust panel of HPV-associated transcripts. So we ended up with 30 transcripts. In red, we can see those that are upregulated in HPV-positive tumors, and the first is CDKN2A, that is the gene encoding for P16, and P16 protein is indeed a proxy of HPV infection. And this gene is also a strong prognostic biomarker for HPV positive tumors and neck in general, because tumors with high expression of CDKN2A have a good prognosis. In green, you see the genes associated with the HPV negative tumors with upregulation in HPV negative tumors, and the top of this list is AKR1C3. This is an enzyme important for the synthesis of cholesterol, of testosterone, and estradiol, and is the top overexpressing HPV negative tumors, and we find it to be a very strong biomarker for negative prognosis. Then we validate our findings by using an independent cohort of more than 100 samples, and we were able to confirm the upregulation of this enzyme in HPV negative tumors compared to HPV positive, and also its association with the survival, not only in the entire cohort, but also in the cohort of HPV positive tumors. As you see in these Kaplan-Meier curves, the red and the orange curves correspond to HPV positive tumors that are stratified according to the expression of AK1RC3. So those that express a higher level of this gene have a worse prognosis, and those with low levels have a better prognosis. So since this enzyme is important for the proliferation not only of hormone-dependent tumors, but also hormone-independent tumors, it is an important and attractive drug target. And there have been already some compounds developed to target this protein. And in particular, we started a collaboration with the University of Torino. We were already collaborating with them in the context of prostate cancer by using inhibitors of this enzyme. But we decided to test it also in cancer cell lines from oropharyngeal tumor that were HPV negative. So we treated these cells with 2 inhibitors of this enzyme, and we found that both compounds were able to decrease the proliferation of these cells, in particular in the cells that were expressing higher levels of the protein. Then we treated the cells with the cisplatin, that is the usual chemotherapeutic agent used for this kind of tumor. And we saw that cisplatin was able to reduce the proliferation of cells, but co-treatment with the compound was not able to improve the efficacy of cisplatin alone. On the other hand, when we pre-treated the cells with the compound, and then after with cisplatin, we were able to improve the effect of cisplatin alone with an enhancement factor of higher than 3. So now we are collaborating with the National Cancer Institute in Milan on this kind of tumor, and still collaborating with the University of Torino to go on with the effort in applying this drug for treating these tumors. And the other aspect that we are facing is the interaction between long non-coding RNAs and microRNAs. So we are analyzing gene expression profiling from the long non-coding RNA point of view and microRNA profiling, because these 2 kind of molecules are very important for the regulation of many physiological processes, and they also interact with one another. Then regarding prostate cancer, here you see that this kind of tumor is the second most frequently diagnosed in worldwide, but the first in Italy. It is also one of the most common causes of cancer-related death, and we work on this tumor since a long time. Diagnosis has been possible by the dosage of the prostate specific antigen that is much used biomarker, but it's not very accurate for diagnosis, because it has many drawbacks. It gives a high percentage of unnecessary biopsies, because it has a high rate of false positives, and may also miss aggressive tumors in men that have low values of the PSA. So as we see from this graph, when PSA is in the so-called gray zone, between 4 and 20, but also 16 nanogram milliliters, we see that this marker is not able to distinguish at all benign prostatic hyperplasia from prostate cancer with low risk or intermediate risk. It could be a marker for distinguishing very high risk tumors, but not this situation. So other techniques such as digital rectal examination are used, but in this technique has a low sensitivity, and multi-parametric NMR is very accurate in detecting clinical significant PSA, but it's labor intensive, time consuming. It requires an experienced radiologist and is also expensive. So final diagnosis requires the use of invasive bioptic sampling. So The idea was to try to combine other biomarkers that can be detected non-invasively, to combine them to PSA to improve the diagnostic path of prostate cancer. So in the age of liquid biopsies, cell-free microRNAs can represent ideal candidates since they are released in broad body fluids, they are stable, and the extracellular release of these circulating microRNAs can be a way to reduce the intracellular level of the microRNAs that have a tumor suppression function, can also function as a paracrine signal to mediate the interaction between cancer cells and cells of the tumor microenvironment, can inhibit the translation of the target mRNAs in neighboring cells. And the evaluation of these molecules in plasma can be useful both for diagnostic purposes, but also for prediction of prognosis, because secretory miRNAs from cancer cells can also be function molecules at distant organs. So we started a study with the aim of combining PSA with other more specific biomarkers that could be detected non-invasively through a blood sampling to better manage prostate cancer detection. We started with discovery phase by microRNA profiling of 60 of plasma. So we extracted RNA from plasma, circulating RNA, and we analyzed samples from 60 prostate cancer patients, 51 benign prostatic hyperplasia, and 27 healthy donors. So we did the microRNA expression profiling with by analyzing more than 2,000 microRNAs, and we retrieved the differentially expressed microRNAs between prostate cancer samples and controls by using the linear modeling for microarray analysis R package. And then we applied penalized realistic regression to microRNAs, PSA, and age. The aim was to build a classifier able to discriminate between PCA and non-PCA. And for this, we used the GLNet R package. Only 2 microRNAs were selected by the model, microRNA 103 and LET7A, and PSA. So these were the only variables with non-zero coefficient, and were used to build a score to calculate the probability of detecting prostate cancer. Then our final model was a 2-step. So samples with PSA higher than 16 were classified as prostate cancer. Otherwise, if the score was higher than a fixed threshold, the sample was classified as tumor, otherwise as non-tumors. So we wanted to validate our methodology, first technically by using more widespread techniques such as real-time PCR, and we were able to validate our previous finding of overexpression of these microRNAs in prostate tumors with respect to normal controls. We analyzed a small population of 30 samples, 10 PCA, 10 BPH, and 10 normal samples. And so the overexpression was confirmed, and we used the minus delta CT values that were calculated by subtracting from the delta CT, from the threshold cycle of our miRNA, the threshold cycle of exogenous normalizer, that was a microRNA from C. elegans, cell 39, that was used as an exogenous normalizer. It was introduced into the sample before RNA extraction in an equal amount in every sample. So this quantity was calculated for both microRNAs, and we compared the 2 classes. Then we wanted to extend this classifier to an additional 242 independent plasma samples that were consecutively collected at the Molinette Hospital in Torino. And we were able to to find a good result for PSA lower than 4, that is the usual threshold applied to send a person to further examination. So when PSA is higher than 4, normally there are some more examination to verify if the tumor is present or not. So if PSA is under than 4, normally the patient is not sent to other examination, but sometimes there are tumor occurring also with the PSA lower than 4. So in this case, PSA is not sensitive enough to detect tumors. And with our classifier, we were able to increase the sensitivity of PSA in this range. Whereas when PSA was lower than 16 but higher than 4, we were able to identify correctly some of the benign prostatic hyperplasia that PSA is not able to identify. So normally these patients have to go to biopsy to verify if the tumor is present or not. And in this case, they do not have any tumor, but the biopsy is an invasive, is very invasive for patient. And so in this case, we were able to increase the specificity of the PSA. Now we are undergoing a very large multi-center prospective study to validate our findings, and the study consists of recruiting 700 patients, 140 cases and 560 controls. It's a collaborative study between our institution, the Italian Institute for Genomic Medicine, and the Medical University of Vienna, and the Molinati Hospital. And we are not only analyzing PSA and circulating microRNAs, the two microRNAs that I mentioned before, and another microRNAs. We are also analyzing circulating metabolites, because from a metabolomics study that we did for patients with PSA between 4 and 16, we found that some metabolites similar to ceramide, together with the microRNA 5100, are able to better discriminate between prostate tumor and benign prostatic hyperplasia in this gray zone of PSA. And this prospective study is also another part involving the prognostic ability to improve the prognostic path of the prostate cancer. And this part involves the analysis of transcriptomic of cancer tissue and the use of some signature that we previously identified in other studies. And my colleague Paola will speak about one of these.
Hello, everyone. In my presentation, I will first give you an example of transcriptomics-based prediction of prostate cancer prognosis. In this work, we focused on the neuroendocrine transdifferentiation process that mainly occurs as a consequence of selective pressure from anti-androgen therapy for the treatment of prostate cancer, and the resulting neuroendocrine prostate tumors are not very well characterized from a molecular point of view. As a consequence, there is no standard treatment, but the prognosis is very poor. In this work, we addressed this question. So which are the molecular features at the time of surgery that may anticipate the neuroendocrine transformation process of prostate adenocarcinomas? And we focused on transcriptomics data, in particular on protein-coding RNAs, long-non-coding RNAs, and microRNAs. We started with the experimental part using microarray technology, and we evaluated about 75,000 features for each of the 54 prostate cancer samples we had. Data were analyzed using bioconductor, in particular the LIMA package, which uses a linear model approach for data analysis. Samples were divided into classes. the neuroendocrine-like tumors that were characterized by at least one of each of these clinical features, together with high levels of chromogranin A, which is a neuroendocrine marker. And then they were compared to the remaining 32 adenoprostate cancer samples. First of all, we performed a class comparison, and we found that among the genes upregulated in the neuroendocrine-like tumors, there were many neuroendocrine markers, genes involved in castration resistance or expressed in thyroid, neuronal genes, and some genes involved in the estrogen signaling. When you manage larger data sets, an important starting analysis is the PCA, the principal component analysis, which is a dimensionality reduction method that reduces the number of variables, and it takes only the variables that mostly describe the data by avoiding loss of information. From this analysis, there was no a clear separation between the neuroendocrine-like tumors in blue and the adenoprostate cancers in red, either using mRNA and long non-coding RNA data or microRNA data, pointing to the fact that these two classes of samples are very similar from a molecular point of view. Another important analysis when you have to analyze the larger lists of genes is the functional enrichment analysis, which tries to group genes into common processes or pathways, and you can assess this enrichment, the significance of this enrichment through, for example, Fisher-Exact test. And here, for our list of differentially expressed genes, we found that the upregulated ones were mainly involved in regulation of hormone levels, neurological processes, and again, the estrogen signaling pathway, while the downregulated genes were mainly involved in the androgen receptors signaling crosstalk, which was expected because neuroendocrine prostate tumors do not express androgen receptor or very low levels, and then other metabolic processes. We then perform a network analysis to understand how our list of differentially expressed genes interact with each other. We used only direct interactions between the objects, so no additional object was added to the network, and we used validated biological pathways. And again, we found that the most connected hubs, so the most connected proteins, were the estrogen receptor and the androgen receptor. So we drew the network using these two receptors. We can see the highly interconnected network with some proteins that were regulated by both receptors. We then perform many other analyses, but the important thing is that we could validate our results, especially those on the estrogen receptor signaling involvement in this kind of transformation. That was quite new finding for these tumors. By exploiting publicly available gene expression datasets present on the GeoOmnibus Expression Omnibus database. We exploited, for example, xenografts models of neuroendocrine prostate cancer or prostate cancer cell lines with estrogen or androgen perturbation signaling, or organotypic sliced cultures. We finally tried to derive a gene signature able to predict this neuroendocrine transformation. First of all, we tried with the complete list of our differentially genes that was composed by more than 500 genes on an independent data set, and it worked well. In fact, we can see a clear separation of neuroendocrine tumors in blue here on the left from adenoprostate cancer on the right. But we were interested in having a gene signature with a few lists of genes. So we used penalized logistic regression with the ridge penalty, and we came out with an 8 gene signature. And again, we validated this signature on the same independent data set and on another one with good values of sensitivity and specificity. So the take-home message here is that we, thanks to transcriptomic data, we were able to point out the putative role of estrogen signaling pathway in the neuroendocrine transdifferentiation prostate cancer. And we were also able to derive a signature of 8 genes that can be applied to predict neuroendocrine prostate cancer transdifferentiation, starting from localized tumors at the time of diagnosis. Another issue we addressed regards the white bias that genetic research has. It means that genetic studies nowadays are dominated by a subset of white individuals, and this may influence disease diagnosis, disease prognosis, and disease treatment. In this work, we wanted to understand why human population of Black African ancestry have a higher risk of aggressive keratinocytes-derived squamous cell carcinomas that are non-melanoma skin tumors. For doing that, we started from genotyping analysis. We analyzed 62 skin samples from Black and white individuals, and we analyzed more than 50,000 SNPs. Again, we performed a PCA. Here we can find a clear separation, a clear concordance of genotyping and ancestry. Here in orange, we have European people, and here Black people, and there was a good concordance of the genotyping also among the self-reported origins. So here we have East African individuals, in violet West African, and in blue Middle Africa individuals. And on the same samples, we perform transcriptomics analysis. We compare Black versus white people. We obtain a list of differentially expressed genes between the two populations. And those genes were mainly involved in metabolic processes in this case, and especially in the oxidative phosphorylation process. Here we worked on healthy tissue, but we wanted to understand what happened also in tumor tissues. For doing that, we exploited the TCGA database of omics data. In particular, we used the transcriptomic profiles of 520 head and neck squamous cell carcinomas, also in this case dividing white people from Black people. And we ended up with a list of commonly differentially expressed genes in Black versus white individuals, in tumors and healthy keratinocytes. Among this list, the HSD70B7 gene was the top-ranked gene, and so we decided to focus on it for further analysis. We then performed a survival analysis on this data set of head and neck squamous cell carcinomas using the expression of HSD, and we found that high expression of HSD70B7 was associated to a poor prognosis, while low expression of this gene was associated to a better survival, even by stratifying by gender. We then perform a Cox regression model to adjust this survival analysis also for age, gender, and ethnicity. And in all these three cases, the high levels of HSD expression remain associated to a poor survival. We then combined the genomics and transcriptomics data we obtained in the previous slide on healthy keratinocytes to search for EQTL. So EQTL are expression quantitative trait loci, and they are the genetic variants that can explain variation in gene expression levels. So we found 9 EQTL across the loci of HSD70B7 gene. 6 of these 9 SNPs were also validated on the 1000 Genome Project as having different allele frequency in Black versus white individual. And here we can see an example of 2 of these EQTL. We can see, for example, that this AA genotype that is mostly frequently represented in white people corresponds to low levels of HSD70B7 expression, while the GA and the GG genotypes that are more frequently present in Black people are associated with a high level of its expression. These EQTL were also validated through the GTEx portal database that contains EQTL data for many normal tissues. And these 6 SNPs were associated with the differential expression of HSD70B7 in many tissue types, including skin and epithelial tissues. From this work, we can conclude that omics data, transcriptomics and genotyping data in this case allowed us to obtain a candidate gene to work on with the other vintage biological experiments. And we found that Black people have some variants different from white people that lead to high level of HSD expression, and this finally results in higher stemness and oncogenicity potential of keratinocytes that can explain why Black people have more aggressive skin tumors. I want to conclude my presentation by summarizing some of the repositories and the database that are freely available and that contain a large amount of omics data, and that you can use for your own purposes to start, for example, with a biological hypothesis or to validate your data. For example, ArrayExpress and Geo are the 2 main data repositories, and you will find there many transcriptomics data, but also genotyping data and some chipset experiments. Then the GTEx portal, that is a tissue-specific gene expression and regulation database. It collects 54 non-diseased tissue sites across nearly 1,000 individuals, and here you have transcriptomics, genotyping, and EQTL information. The TCGA, as already mentioned, here for tumors tissues, you can play with the TCGA on its website or through the CBio portal or website. You can download the data from Firehose website. Then for the functional elements of the human genome, there is the ENCODE project. Here you have information of chromatin accessibility, chromatin interactions, chromatin modification, and you can interrogate the ENCODE project through the genome browser of the UCSC. And finally, the 1000 Genomes Project, that is a catalog of human genetic variations for more than 2,000 individuals from 26 populations around the globe. And here you can interrogate it using SPS Smart or this tool from the NIH website. I thank you for your attention, and if you have any questions also in the future, you can write us an email. Thank you.
Thank you, Giovanna and Paola. Very interesting presentation. I have some questions, but I will save for the end of our afternoon for the public discussion. So for now, thank you. And I move to the second speakers, the second set of speakers, Barbara Pioselli and Maria Laura Faietti from the Chiesi Pharmaceutics, and I will
okay, can you see my screen?
Yes, fine.
Okay. Fine.
The registration is active.
Good afternoon to everyone. I want to thank you, the organizing committee, for inviting me to this webinar. Today, I will give you a brief introduction on metabolomics. Then I will move to show you the most important step to plan a good experimental design. And I will finish with a few slides on translational metabolomics. Let's start with, Let's start with omics. The map of our genetic self was an incredible milestone in scientific history. And for omics, it was the Big Bang that gave birth to an ever-expanding universe of polar analysis. Today, that universe is almost unrecognizable from the one we inhabited at the turn of the millennium, and its associated applications are booming too. Many areas of research can be classified as omics. Examples include genomics, transcriptomics, omics, and metabolomics, with lipidomics, glycomics, which correspond to global analysis of genes, RNA, proteins, and small molecules. In this slide, you can see the number of listed publications in PubMed over time, using the corresponding omic discipline name as the keyword. Genomics is the predecessor of all other omics technologies, making it the most mature and far-reaching omics field. As you can see in panel A, the number, sorry for the phone, a moment. So in this slide, you can see the number of listed publications in PubMed over time using the corresponding omic discipline name as the keyword. Genomics is the predecessor of all other omics technologies, making it the most mature and far-reaching omics field. As you can see in panel A, the number of articles published on genomics, colored in gray, and proteomics in light blue, seems to have plateaued since 2017. Meanwhile, the minor omics disciplines, transcriptomics, metabolomics, and glycomics, continue to grow exponentially. Especially lipidomics has undergone an explosion of publications in 2013 and continues to experience a sustained and impressive growth, as you can see in panel B. But what are metabolomics, metabolites, metabolism, and metabolome? The term metabolomics was introduced over 20 years ago, and since then, remarkable improvements have been made to the analytical platforms and data analysis pipeline. In the beginning, metabolomics was described as a tool for functional genomics that can be used for the analysis of all metabolites produced by a cell or a system. However, we are still a long way for getting comprehensive coverage of all metabolites, even though drastic developments have already taken place. Generally, metabolomics can provide insight on the biochemistry underpinning response of an organism to an external or internal alteration. Many metabolomic studies are focused on finding markers for diagnosis of existing conditions or prognosis of physiological conditions that may not be clinically evident. Metabolomic methodologies fall into two distinct groups: untargeted metabolomics and targeted metabolomics. Untargeted metabolomics can analyze metabolites derived from the organism comprehensively and systematically. It is an unbiased metabolomics analysis that can discover new biomarkers. The data are acquired without a priori knowledge of a biologically interesting metabolites. Targeted metabolomics is the study and quantitation of specific metabolites. The metabolite's identity must be known, and no further metabolite identification is required. Both has their advantages and disadvantages and are often used in combination. First, the untargeted approach, followed by the targeted approach for the discovery and determination of differential metabolites. Metabolites are low-weight compounds with a molecular weight generally less than 1,500 Daltons, that are not protein or peptides, with the exception of glutathione, that is a tripeptide but has a metabolic function, and can be defined by physicochemical properties, chemical structure, and biological function. These small molecules perform function or tasks necessary for cell growth, inhibition, and stimulation. Among the metabolites, we can find carbohydrates, nucleotides, organic acid, amino acid, and lipids. We can observe endogenous metabolites produced by the organism itself, like fatty acid, organic acid, sugar, and vitamins, and exogenous metabolites external from the organism, like the drugs. All the metabolites are interconnected in the metabolic network. The metabolic network is more than the sum of its parts, and a deep understanding of metabolism requires that all of the uncharacterized complexities are dissected. The crosstalk that occurs between the different parts of metabolism plays a crucial role in metabolic network. Realization of metabolic crosstalk leads to a more complete understanding of the metabolic network in the cell, tissue, or biofluid. And this knowledge is widely applicable for basic and applied sciences. The metabolic network describes the interaction and interplay between the various metabolic pathways and can be altered by environmental factors, disease, or diets. For examples, as you can see in the figure on the right, in low glucose condition, ATP production via fatty acid oxidation is increased. This is called the Randle cycle, also known as the glucose fatty acid cycle, a metabolic process involving the competition of glucose and fatty acid for substrates. This mechanism allows preserving glucose for tissue that use fatty acid and depend on glucose for energy, and it seems to play a role in explaining type 2 diabetes and insulin resistance. The metabolome represents the complete set of metabolites in a biological cell, tissue, organ, or organism, which are the final product of gene transcription and protein translation, and so closely define the phenotype. The dynamics of the human body are mirrored in the metabolome. For example, after eating a meal, the quantitative and the qualitative composition of blood and urine metabolomes change in a timescale of minutes to. One of the major challenges in metabolomics is measuring the complete metabolome of biological entity using a analysis method. Why it is so hard? The Human Metabolome Database, called HMDB, which is a freely available database containing detailed information about small molecules found in the human body, contains more than 2,000 of metabolite entries, including both water-soluble and lipid-soluble metabolites. Metabolites are tissue, cell, or biofluid specific. In fact, the metabolome changes quantitatively and qualitatively. Just to give you an example, the relative distribution of lipids in plasma sample is sterolipids, about 31%, triacylglycerol lipids, 43%, and triacyl phospholipids, 43%. Whereas the fluid that covers the lungs, called surfactant, contains mainly phospholipids that cover 80% of total lipids. Measuring the metabolome of a tissue, a biofluid, or a cell can be complex, because it contains a chemically diverse range of metabolites with concentration that vary orders of magnitude, from gram to liter to less nanogram to liter. Moreover, we can face with both polar and non-polar metabolites. Metabolites are also different in terms of their stability, as they can be oxygen or light sensitive, and they do have very different turnover rates inside the cell. And so, we can measure the metabolome in a single run and with one platform. At present, the answer is no. Given the complexity of measuring the metabolome, in order to plan a successful metabolomics experiment, one has to start with a robust experimental design. This is often neglected, but the high dimensionality of data means that it needs special attention. In the design of experiment, the experimenter is often interested in the effect of some process or intervention, like the treatment, on some objects, the experimental units, which may be people, parts of people, group of people, plant, animals, materials. It is easy to discover metabolic changes, but are these directly related to the biological question, or are they related to confounding factors? In other words, you have to plan a study to ensure that the variation related to the biological observation are greater than the variation introduced by performing the study. Again, these are called confounding factors. A good experimental design includes careful consideration of the samples or sample types, the selection or choice of controls, the level of control over external variables, the required quantities of the samples, the number of biological and technical replicates, and the preparation and storage of the samples. Additionally, other important factors that need to be considered while designing a good multi-omics experiment are the meta-information collected about the sample, the selection of omics method, and use the requirements for data storage. Lastly, it is of paramount importance to ensure that sufficient time and financial resources are available to carry out the experiments. Always remember that a good experimental design is to avoid that your efforts, your time, last but not least your money, are used to produce results that do not answer your biological question. Remember to discuss your experimental design with a biostatistician before starting out, because as Sir Ronald Elimer Fisher said, to call in the biostatist after the experiment is done may be no more than asking him to perform a postmortem examination. He may only be able to say what the experiment died for. Some experimental processes to consider during experimental design are: what is your research question, sample size and type, sampling and sample extraction strategy, analytical technologies, collection of metadata, confounding factors, randomization, analysis strategies, biological interpretation, validation, and many others. First of all, you need to say what is your research question. It is important to know what biological and research question you are asking. Metabolomic studies of the human population usually fall into three basic categories: predictive biomarker discovery or prognostic test, with the aim in translation into diagnostic prognostic test, pathogenesis studies with the aim to uncover mechanism of disease, association studies with the aim to find correlation between human metabolome and factors such as demographic, lifestyle, and physiological factors. The first area and core of any metabolics experiment is sample collection. So just few quick considerations on sample size, sample type, sample collection, and sample normalization. The sample size depends on the environment of the experiment. If you are planning to work with cells or animal models with the same type, you will have a relatively small biological variance. In this case, the sample size can be limited to 6 or better 10 replicates per group. However, humans live in a non-controlled environment where food, lifestyle, age, and gender are not controlled for, and so a greater number of samples are required to determine this variability. This will lead to high interclass variability, and your sample size must be larger, but the number of samples will depend on the question being asked. In a metabolomic study, you can choose among different sample types, such as culture cell and media, biofluids like plasma, serum, urine, tracheal aspirates, and tissue. The choice of the sample type will depend on your biological question, because if you're looking for a biomarkers of kidney transplant failure, you have to look at urine, that is the biological fluid close to the organ you are looking at. And at the same time, if you are looking at the effect of a drug for lung respiratory distress, you have to sample the lung epithelial lining fluid. However, the choice of sample type depends also on sample availability, on number of samples required, and on the ease of sample preparation. You can obtain easily serial sample of urine, but it is difficult to obtain close sample of blood, especially in fragile categories like infants and pregnant women. It is useful to have a standard operating procedure for pre-analytical handling of samples before starting a metabolomic study, especially if you are planning a multi-center study. Probably most of the primary care facilities do not have access to expensive instrumentation, centrifuge at low temperature and quenching protocols to inhibit the degradation of metabolites. In this case, dry ice frozen transport of clinical samples to the main laboratory could be an option, a bit not cheap. Aliquoting the samples should also be considered whenever possible. This important step will avoid repeat freeze-thaw cycles that lead to progressive loss in sample quality. Finally, long-term storage at -80 degrees or less is recommended before analysis. Moreover, always remember to collect control samples. This is essential for a good study. The choice of time of collection of a sample is a crucial step for a successful metabolomic study and will depend on the research under examination. Nutritional status on the experimental subject greatly influence the circulating urinary, fecal, and tissue metabolomes. In this case, your standard operating procedure, you must detail the time of collection. Circadian rhythms govern a large variety of behavioral, physiological, and metabolic processes. Recent advances reveal that a very large fraction of mammalian metabolism undergo circadian oscillation. Many metabolic pathways are under circadian control and in turn may feedback to the clock system to assist circadian timekeeping. In more than 40% of the serum metabolome and 45% of the liver metabolome has been shown to be sensitive to time. Most metabolomic studies are based on non-invasive or minimally invasive samples types, such as blood, urine, and feces. However, tissues are also important as the tissue represents the first place where the metabolic changes owing to disease taking place. More tissues are non-homogeneous. For example, the liver has 5 different lobes, and within them, the portal and periportal vascular regions are known to display different levels of some enzymatic system, such as those involving glycolysis and gluconeogenesis. Even when the tissue is composed on the same cell type, regional differences in composition may exist. Such factors may result in increased biological variability, which should be taken into account during sample. Sample normalization is a critical step in both qualitative and quantitative metabolomics, but there is no unified method to do it because it depends on sample type. If you think, for example, in a biofluid such as urine, the total concentration of metabolites can differ by more than 14-fold. Even for the same individual, the urine concentration can vary greatly, depending on water intake, diet, and excretion. Because urine concentration can be influenced by external factors, in clinical field, this varying factors is often controlled by using a reference analyte presenting all urine samples. Creatinine is the most common reference, and the assumption is that creatinine concentration reflects the urine concentration. The sample concentration of feces, bronchial fluid, lavage, etc. can vary greatly also. The cell extract of tissue can also have different total amounts of metabolites depending on several factors, including the number of cells, the lysis efficiencies, the composition of cell, and the cell density in tissue for tissue sample. The differences can be due to the sample concentration differences, the non-standard differences caused by a true differentiating factor such as a biological event, or a combination of the two. Because we wanted to determine the metabolite concentration changes caused only by a biological event, or by a non-biological factor, sample normalization prior to analyzing all the individual samples should be considered as a critical and necessary step in the overall metabolomics profiling workflow. In this slide, you can see why sample normalization is important. This is an analysis of the lipid profile bronchoalveolar lavage fluid taken from human with bronchopulmonary dysplasia and human with normal lung. Bronchoalveolar lavage is a minimally invasive procedure that involves instillation of sterile normal saline into a subsegment of the lung, followed by suction and collection of the instillation for analysis. This procedure leads to sample with different dilution, and so are the metabolites in the sample. In the top of the slide, you can see the volcanic of the metabolites obtained before the procedures of the sample normalization, and on the bottom, those obtained after sample normalization. And see that there is a huge difference. This is a post-process normalization based on urea concentration. However, it is better to do a pre-process normalization to avoid differences in sample recovery during the preparation step and ionization efficiency during the analysis. But let's move to the analytical technologies. Metabolomics use a wide variety of analytical instruments that vary in terms of their sensitivity, accuracy, and range of coverage. Nuclear magnetic resonance, it is often chosen for its reliability and utility in absolute quantitation. However, NMR is relatively insensitive and is limited to measuring substances in micromolar to millimolar concentration. A spectrometry platform, especially LC-MS, can detect metabolites at nanomolar to picomolar concentration, allowing a much higher number of metabolites to be detected. However, MS instruments are prone to frequent breakdowns, and relative to an NMR, it is often difficult to quantify concentration via MS technique. Gas chromatography MS, GC-MS, is less sensitive than liquid chromatography, is generally more robust and more reproducible. As a result, GC-MS can sometimes be used to quantify the metabolome with higher precision and reproducibility than either NMR or LC-MS. Aramadurian said that there is no one magic tool that can capture the diversity of composition and concentration which is present in a single sample. It really depends on the question you are asking for. You need an array of tools available. Confounding factors are variables that are not directly being studied, which can cause inaccuracy or bias in the measurement or prediction of outcome of interest. The collection of metadata in a design is important as it allows the researcher to assess whether any confounding factors detrimental to the study are present. Many factors are known to influence the composition of the metabolome, and these confounders should be minimized or eliminated to provide an easier data analysis procedure. Most typical confounding factors are age, gender, drug intake, BMI, sample age, and sample size. Nowadays, a lot of studies have provided evidence that age and sex influence the metabolome. This is a figure taken from an article published in 2009. The figure shows the effect of a 10-year increase in age on the top 100 metabolites most strongly influenced by age. Positive values indicate the amount of metabolite increase over 10 years, whereas negative values indicate the amount of metabolites decrease over 10 years. The authors concluded that 57% of metabolites assessed significantly changed with age, 84% of which increased with age. Of the total 34 steroid lipids tested, 29 significantly decreased with age, 2 an androgenic steroid and cortisol significantly increased with age. Higher levels of the most fatty acid lipids were associated with increased age, including 13 out of 14 long-chain fatty acid, 28 out of 34 acid carnitines, and 42 out of 78 other fatty acids. Higher levels of sphingolipids tended to be associated with increased age. In this slide, you see the metabolites related to gender. Positive values indicate that the metabolite was higher in women, whereas negative values indicate that the metabolite was higher in men. 63% of metabolites assessed significantly different by sex, with the slight majority, 5% found in lower levels in women. On the metabolites associated with sex, most of them are also associated with age. 29 sterolipids were found in significantly lower levels in women, with the exception of 2 corticosteroids including cortisol and corticosterone, which were found in higher levels in women. Androgenic steroids constitute the three metabolites most strongly associated with sex, but some lipids are higher in women. From these two slides, you can understand how important the construction of your study group is, especially when you need to build your control group that must be similar to your study group, at least in age and in gender. Regarding the importance of drugs as confounding factors, I want to present briefly a study we did a few years ago in infants with exposition of great arteries, congenital heart defect. These newborns undergone surgery in the first week of life to correct the cardiac defect. With this study, we aimed at seeing if the surgery would affect the urinary metabolome of these patients. We collected urine samples before and after the surgery and analyzed them. The problem was the drug intake because these infants took about 25 drugs during the surgery, and this is a big or a huge confounding factor. We searched all these drugs and the related metabolites in a pool of urine samples. Then we built a list and we used it to clean our results. It was a long work that lasted 2 months only to find the drug metabolites in the biological sample, but was the only way to obtain data influenced by the surgery and not by the surgery past the drugs. Although there are many tools for analyzing and interpreting the massive amount of data of an untargeted metabolomic experiment, it is still considered as a bottleneck of omics technologies. This is a snapshot of an output of the analysis of an untargeted metabolomic experiment done in cell. As you can see, we have a huge amount of data. In this table are listed more than 1,000 compounds, some with a name and an associated formula, some with only a formula, and some with only the molecular weight. Listed here are those provided by the comparison with online fragmentation database spectral libraries like Matlin, MZCloud, or MZVault. But although the development of these libraries have provided a benchmark for identification of a wide range of metabolites, more bioinformatics tools are required to enhance automated spectral analysis and metabolite identification. But how many metabolites will be identified in a global untargeted metabolomics study? This is a difficult question to answer. We typically observe between 500 and 2,000 compounds, depending on sample type. It is important to note that there is a range of annotation confidences assigned to each detected compound. We use a classification system to describe the annotation confidence. Briefly, more experimental evidence like fragmentation data, collision time, collision cross-section, reference standard, etc., enables a higher confidence metabolite identification assignment, while some compounds be identified with the comparison of a standard, and so they are on level 1. Others may be reported in groups, multiple candidate annotation level 3, owing to the fact that a lot of metabolites are isomeric and metabolite databases are incomplete. For lipids identification, the spectra fragmentation library are scarce, as lipids is Genomics. So we are building our in-house library to reduce the number of candidates and for increase the confidence of candidates when possible. However, the metabolites with a good identification are usually 10 to 12% of those reported by the program. Now you have your list of identified metabolites, but how are they connected? Are they referring to a pathway? While there are many excellent tools for multi-omics integration and visualization, there is currently still room for improvement. One area where significant work is needed lies in the availability of pathway databases. Pathway databases provide biological context while at the same time providing clear links between genes, protein, and metabolites. Metaseq, Bioseq, Metabolica, and Reactome are freely available and widely used pathway databases. However, they are limited in portraying what is known about biological pathways or molecular processes. While all the databases emphasize metabolic pathways, they were all developed before the advent of metabolomics, and thus lack of some of the new insight that these technologies bring. Although targeted and untargeted metabolomics workflow have many advantages over classical analytical chemistry, there is still many limitations and challenges that need to be addressed in the advancement of this new omics field. As a data and question-driven approach, metabolomics has shown high potential in hypothesis generation and marker discovery. However, the current spotlight on metabolomics may not last long if we do not drive the field towards applied research that would have a direct positive impact on the advancement of industries and welfare end users. Omics is broadly defined as applied research which aims to turn the results of an omics experiment into a useful turnkey product. High-throughput omics technologies may allow more informative characterization of disease to better predict both individual patient clinical course, the degree of benefit he/she may derive from new and existing therapies. The potential for detailed characterization of disease has been met with great enthusiasm in oncology, where the heterogeneous character of malignant disease has long presented challenges. However, despite widespread use of these technologies in both preclinical and retrospective studies, it has proven more difficult than expected to translate their promise into clinical use tests that can be used to guide management decisions. In the case of Duke University, a series of papers appearing in major journals such as Nature Medicine and the New England Journal of Medicine shows that the gene activity in a patient tumor cell could be used to determine which chemotherapy drugs would be the most effective for that patient. This capability will enable significantly advances in cancer treatment. Soon after the first papers were published, Kit Baggerly, Kevin Coombs, and Jim Wong, bioinformaticians at the Cancer Center of the University of Texas, began working to replicate the results. They immediately encountered difficulties using the data made publicly available with the paper and began communicating with the researcher who did the work. Data provided by the Duke team contain numerous anomalies and obvious errors, making it impossible to replicate or verify the results. The papers has been retracted. Following this and other cases involving premature use of omics-based tests in cancer clinical trials, the National Cancer Institute requested the Institute of Medicine establish a committee to recommend ways to strengthen omics-based test development and evaluation. This report identified best practices to enhance development, evaluation, and translation of omics-based tests. The Institute of Medicine defined an omics-based test as an assay composed of or derived from multiple molecular measurement and interpreted by a specified computational model to produce a clinical actionable result. Metabolomics is increasingly being applied towards the identification of biomarkers for disease diagnosis, prognosis, and risk prediction. Application of metabolomics in biomedical research has achieved discovery of variety of biomarkers for disease such as risk to diabetes, heart disease, and cancer, while also making progress for a variety of drugs. Unfortunately, among the many published metabolomic studies focusing on biomarker discovery, there is very little consistency and relatively little rigor in how researchers select, assess, or report their candidate biomarkers. In this sense alone, there have been more than 1,600 publications that claim to have discovered a biomarker using a metabolomics approach, with nearly half of all papers survive. Although there are some exceptions, most of these researchers fail to acquire enough statistical power due to a limited size, less than 100 subjects, and almost none repeat the analysis in a further cohort and thus fail to demonstrate a lack of biomarker utility. In particular, few studies report any measure of sensitivity, specificity, or provide severe operator characteristic curves with associated kinds of intervals. For metabolomics, in the past 5 years, quantitative metabolite measurements have enabled the translation of less than 100 chemical tests to the clinic. Clinics say that data does not equal information. Information does not equal knowledge, and most importantly of all, knowledge does not equal wisdom. We have oceans of data, rivers of information, mud puddles of knowledge, and the odd drop of wisdom. In this slide, you can see a schematic representation of the major steps for metabolomics biomarker discovery. This initially starts with a discovery phase, which involves in-depth metabolomic assessment. This tends to be done in relatively small cohorts, about 100 subjects. Following this, a pre-validation phase then repeats this untargeted metabolomics assessment in a patient control cohort, another 100 patients, but preferably from a geographically distinct area from the first discovery phase. Following this, there is an analytical development phase for the assessment of biomarkers discovered using lower cost technologies. This represents a shift from mass spectrometry or NMR spectroscopy to targeted chromatography or direct measurement, using, for example, lateral flow devices. Finally, using the faster and cheaper technology, there is a validation phase in a large patient cohort from 10,000 to 100,000 patients to assess the robustness of the biomarkers discovered. The focus of biomarker discovery should not only be for pathological cures, but also for preventive screening of healthy individuals, as earlier biomarkers may be useful in directing dietary and lifestyle changes prior to more radical surgical treatment. Just to conclude, in order to successfully translate the results of a metabolomics experiment, you need to start with an excellent experimental design, continue with data acquisition, data mining and interpretation, finish with the validation of one or more candidate biomarkers, better more candidates, and the inappropriate conduct of these steps, which is translation of metabolomics into clinical and industrial application. Thank you for your attention. I'm finished.
Thank you, Manuela. It's a very comprehensive presentation and I think stimulating for questions. So we are at the end of a session and it's open for floor discussion. I have from the webinar some general question. Just I read from the chat. One is for Giovanna. What omics data, in general, genomics, transcriptomics, proteomics, epigenomics, what are currently most effective for cancer detection and prediction of prognosis? And another question to this one is, what do you think about the multi-omics approach?
Okay, so I start with the second. I think that multi-omics approach is very important, especially to better understand the feature of cancer, of a disease in general, but in particular for cancer. And so it's also useful to integrate different aspects, different levels of alteration that can occur, for example, at DNA level or RNA level or protein, to better understand why, for example, a specific protein is overexpressed or underexpressed in tumors. So to understand the reason, the mechanism why it is like this. But when and so applying omics approaches is very important especially in the phase of discovery of the biomarkers. But when we have to build a classifier or to have some specific biomarkers that have to be analyzed practically in a clinical context, we needed to have very few biomarkers that should be easily detectable in a non-invasive way. So and also with cheap technologies. So we needed to, we can, for example, combine biomarkers that come from different levels of analysis, but they need to be few and not necessarily be interconnected, because maybe they can complement each other and give a more comprehensive information about, for example, the presence of the tumor. In terms of prediction of prognosis, for example, in this case, in most cases, the tissue is available. For example, bioptic or surgical tissue is available, so it's possible to apply, again, in a discovery phase, a high throughput methodology, but then maybe to derive a small signature that can be applied into the clinics, or for example, starting from transcriptomics, to be able to derive a protein marker that can be easily detectable, for example, by immunohistochemistry in a pathology department. So I think it's important to integrate the multi-level to better understand the disease. But then when it comes to have a specific tool that can be translatable into the clinics, we have to deal with a small number and easily detectable molecules.
Okay, thanks. Waiting for other comments. I have some question for you and Paola. 3 questions. One is regarding developing a diagnostic score. When we move from the development phase to the proposal for a practical use of a test, we usually perform a decision analysis, balancing the utilities and costs. Are there examples in prostate cancer field? And do you face this issue in some part of your work?
Yes, for prostate cancer, there is a lot of ongoing research and many tools that have been proposed, but none of them has already been applied into a clinical context, because there is still not a real screening tool for prostate cancer. For example, for breast cancer or cervical cancer, we can have a screening, but for prostate, not yet. So there are many efforts in this field, and of course we need to have something that is easily applicable to many, especially in a specific age range. So this is why we decided to focus in the 50-69 age range, because, for example, after 70 years old, okay, it's important to detect the tumor, but most of the tumors are indolent, and so even if we detect the tumor, we can do overdiagnosis and overtreatment to people that may live with this tumor without having any particular problem.
Yes,
So,
yes, it's a main problem in prostate cancer screening.
yes. So,
Please.
no, okay, it's okay.
My second question is a minor one. You mentioned the use of a ridge penalty, which I personally like. So I wish to ask you why you choose it with regard to the lasso penalty.
Yes, we chose it because we started already from a small number of features to validate. So we didn't want to make a feature selection,
yeah,
but only to create a score. And so since we obtained these 8 features from the intersection with the independent data sets, we decided to use the ridge penalty.
so you maintain some impact for almost all of the regressors.
Yes, 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 yes.
And the last one is regarding, it's very interesting, the Wyatt bias.
Yeah.
Because most of the biobank are based on a subset of a population with
Yes.
some specific characteristics. But this is a selection bias. While in your analysis on ethnicity, you address something like confounding or potentially an effect modification. So it's only indirect the connection with a white bias.
Yes, yes.
So I lost the screen, just, okay. Luca, do you want to make a comment on?
Yes, as I was saying in my presentation, the reference for omics study, it's a problem. I mean, for general purpose studies, without complex questions, they are fine. They work well. They worked and they work well. For genetic and genomes, the technique is mature. Also, there's a lot of people working on it. And sorry.
Oh.
Quarantined. So
yeah. I know.
Everyone in the same situation.
Yeah.
So, so the problem, the reference genomes are okay. A lot of people are working on it. As Manuela can say to you, there's a lot of problem for proteomics and metabolic studies. Proteomics, not a lot, but for metabolomics reference, it's quite a mess. For example, in a metabolomic study, you can recognize from the features if you are very lucky, from 10 to 20% of the metabolites you measured. So you know the number, you know you have the number in your instrument, but you cannot link the number to a name. So this is a problem. And also, it's a problem for transcriptomics. For example, the discovery of the CRISPR-Cas9 system that gave us a Nobel Prize also for Jennifer Doudna. They had to postpone the publication of the first paper because the reference for the mRNA was wrong in the database. So they had to sequence everything and redo the database. So yes, it's a problem. also because we don't have to forget that databases, omics studies are possible because non-omics studies, because databases are built by single experiments. So a single gene is characterized, sequence, and everything. So omics builds on non-omics studies. And we will see. So artificial intelligence can help, can do the most tedious jobs, and then we have only to check. But checks are essential.
Okay, thanks. Some other question. Let's see. I have one for Barbara and Maria Laura about targeted analysis. How much prior knowledge could limit new discoveries, in especially with regard to drug discoveries?
I don't really know if Barbara is already with us.
Okay, I recover my so both Maria Laura Faietti and Barbara Pioselli left. So Luca, what's your opinion? And maybe also Manuela and Giovanna and Paola. If you search for new drugs and you rely on targeted analysis, you probably may
you can use Metabolomics to see the effect of a new drug or a treatment, but to develop a new drug, so I don't really know. You can try different drugs of modern animals and see the output and see if the metabolism changes after the drug, but in drug development, I don't know.
Yes, I was surprised of having all the full spectrum from unsupervised to targeted analysis for drug discoveries. There is another question in the chat, Luca.
Yeah, I reported some question that I got from someone that has left, so I put it there.
Yes, please read it.
How to measure real clinical impact and cost effectiveness of prospective omics approaches, and how will intellectual property regulation apply to knowledge regarding omics data and methods and techniques developed thanks to that knowledge. So for I can try to answer the question. So to measure the clinical impact, we will be very happy to know how to do that. Yeah, cost effectiveness, it's simpler to calculate, also because for now, even if the whole genome of all exome cost is lower and still lowering, it's still quite high to apply to every patient or in an untargeted way. I mean, untargeted in this way for patients. And so, yeah, the most of the cost are the analysis, the actual analysis is in the lab. And yeah, you can start from there. I think you can start from the cost of the lab, and if you can scale up. For example, genomics can be scaled up very efficiently, but not proteomics and not metabolomics. It's very difficult to scale metabolomics and proteomics, so the cost will rise will rise a lot. Intellectual property regulation for omics data. So I think that the question is linked more or less, as you can see from the open source software. So you can regulate knowledge in the sense of data, but you can regulate and you can have intellectual property on how you handle data. So you can patent a process, but you can't patent the data. So in the omics data, since results are so variable and so depending on the analysis, that means that the data analysis of omics data is itself an experiment. So there's not a pipeline, a defined pipeline for analysis. So it's an experiment itself. And for every journal that publish proteomics, metabolomics, and I think genetics, but I don't know how is the panorama of this, they require the data because it's an integral part of the study, data and the data analysis. It's very difficult to find someone that didn't share the original data for omics study. This is a very good thing, and yeah, in this way you can patent the paper, but you can't patent the data. So I think, yeah, we can write a book on this, but
sure. But the question on the intellectual property has several different answers. It depends. We can first discuss not in terms of like a patent, but in a scientific term. And this is as a problem with open science architecture, which is implicit when you have to provide the original data together with the paper and the program code. So it's linked to the reproducibility problem of this kind of analysis and the inherent acceptance of the open science structure. So you should be aware that you are working openly, and each researcher contributes with a bit, and your data are shared using some regulation, of course. But the regulation and in some sense protect large research consortia, not the single researcher, the large consortia and the large institution, the large scientific bodies. The other aspect is more interesting from the bioethical side. Because on the ultimate, you are using biological specimens which are conferred by a donor, and the donor still maintains some right on the materials. So there is several cases in the Supreme Court in the US, and one was interesting because it makes recognition to the intellectual property of a researcher, which was considered in that case superior to the right of a donor. For the example is simple: you develop a diagnostic test, you patent the test, and you receive money. Should the donor receive money as well as the researcher who developed the diagnostic test? This is one of a question. So there are several streams of discussion underlying this, and still are not completely resolved. One is the side I can cite, the open science, and on the other side, the question of the right of the donors in all the study in which there is biological materials to be considered. Giovanna, Ileana, Paola, do you want to comment?
Just a quick question for Luca, if it's possible. Luca, in this case, is deep learning driving the change as we talk about methodology?
So, I attended an essay seminar last week with our PhD students on deep learning in genomics. So they are doing some steps forward, but not for now. So yeah, also because it's connected for the reference, because we don't know the reference. It's discovery, it's question generating, and also because you are interested, they were using it for mutation for genomic-wide association studies. So they encoded mutation as 1 and non-mutation as 0. And so they put all this stuff in the neural network. And so one of the problems for them was to derive the causal motif of this variation, because the answer was, is this mutation linked to a disease? The answer from the black box is yes or no, but you cannot publish something that is say yes or no. And so they used something very neat, that techniques that goes backwards. So after you developed the net, this technique go backwards from your results to your data and tells you what was the thread of the network. So you can have a model. It's not a black box, but for now the application are more in a very, very early stage of experimentation. Yeah, because connecting to the problem before, there's the problem of the reference. By the way, artificial intelligence was applied very successfully with AlphaFold to generate the reference. Because from a sequence, it generates a protein. So you can have a reference in this way. I think that probably is this the way. So AI is going to be a reference creator rather than a mechanism to analyze the data, but never know.
Thank you.
I sketched also in my notebook a question for Manuela about potential cost-benefit analysis of metabolomics for diagnostic biomarkers discovery. So in part, you already answered to this.
I haven't heard the question.
Oh, excuse
Can
me.
you repeat?
It seems to me that the funnel is very small at the end. So you did a lot of analysis on a lot of metabolites and molecules and so on. But at the end, the number of new diagnostic tests which were proposed as very small.
Yes.
So in terms of cost benefit, it's worth what is the economics and the...
It is true. There are a lot of study of biomarker discovery that does not translate to the validation phase, and it did not a clinical test that is useful. It is why you have to plan your experimental design from the beginning. If you want to only do a biomarker discovery experiment, you know that you have to spend not so much money, but if you plan to lead to a clinical test, you have to plan a lot of money and a lot of time to spend to do all the process, because you have to start with the experiment process with the pre-validation in about 100 patients. Then you have to validate your biomarker of panel biomarker. It is better a biomarker in another validation cohort that is almost 100 patients from a different geographical area. And you have to translate your biomarker panel from your NMR or CMS that are cost instrument to a targeted instrument like a triple quadruple or two devices that is more cheaper. And you have to date in a bigger cohort that has about 10,000 to 100,000 people. So the process is long. It depends on how do you want to reach? And the cost is different, obviously.
Okay, if you have other comments, questions, suggestions, criticism, please, we have last minute.
I report also, so Barbara is very sorry that she couldn't attend, but if you have question for her, she will be happy to answer extensively by email. You can email us, whatever,
okay,
or Professor Biggeri, and we will forward it to Barbara and his team. Feel free to ask us.
Okay, this is all for all the attendees. So it's okay. I wish to thank Giovanna, Paola, and Manuela especially for having attended, and I also participated to a floor discussion and to this initiative of the University of Padova, which I think is very interesting for all the PhD students of the university. So, and I hope the people of the audience will be satisfied, would be satisfied. We need some evaluation after. Thank you to all. Thank you again for the speakers and for Luca for having organized the session so brilliantly.
Thank you. Thank you very much.
Thank you for inviting us.
Thank you for inviting.
Goodbye.
Good quarantine to all.
Thank you.
Say bye.
Bye.
Bye.